All right, we're live. Sorry, everyone, about the technical difficulties. Um, we're learning about some of the limitations of this software platform as we go. So thanks for bearing with us. Hopefully you all got the
Thank
note to
you.
come over into this session. At the end of this session, what we'll do is we'll sort of clean everything up, rename this one, just make sure that all the information is still organized so that you can still come back and access the webcasts in order. So going back to our regular introduction, hi everyone. Welcome back to today's session of the Escarpment Labs webinar series. We're a yeast producer based in Guelph, Ontario. We work with breweries all over the world to solve fermentation problems and create new flavor opportunities. I'm Richard Priest, co-founder of Escarpment Labs. I'll be the moderator for this webinar. If you're joining us live, please tell us who you are and what you've been doing to pass the time in the last week, maybe what beer you're drinking. Not much else to do. The Crowdcast platform is an excellent way to get conversations started, and community is something we need right now. Today, our topic is Yeast Basics Part 2, Yeast Flavour Production. I'd like to introduce today's presenter. Just like last time, we got Nate Ferguson, co-founder of Escarpment Laboratories, professor at the Niagara College Brewing Program. Nate graduated from the University of Guelph with a degree in biochemistry, and he's a treasure trove of knowledge in all things fermentation. For those that are joining us live, feel free to start a discussion on the Crowdcast platform, ask questions and upvote questions. I'll do my best to monitor the discussion and follow up with our speaker if needed. For those that are tuning in after the live cast, thanks for checking out what we have to offer. You can still ask questions as well, and we'll try to get to everything in the Q&A session at the very end. We will provide a recorded version of the webinar. The first session is still available here on Crowdcast and also up on YouTube. And then again, if you have questions and we don't get to them during the actual series, we're going to do a full Q&A session at the very end as well and try to answer all of your questions. Without further ado, I want to give Nate the floor to tell us all about Yeast Basics Part 2, Yeast Flavour Production.
All right, thanks, Richard. We'll just get this presentation loaded up here. And we're off. So thanks, everyone, for joining this. Just to kind of give everyone an idea as to where we are in the large scheme of things. So last week, or early this week, on March 23rd, we went through the basics of what yeast cells does, with really focus on trying to get a better kind of hedonic idea of what yeast cells are and how we actually work with them. Now, today, we're doing that classic kind of deep dive into the metabolism of the organism to better understand how it actually produces the flavors. Now, we are going to be comparing this a bit more, or looking at this is probably a better way to say it, as an organism and not just as a series of biochemical reactions. And that's going to be a really big difference, and I think something that's going to help everyone when it comes to actually understanding these organisms. So with that, today we're going to talk about what flavor yeast cells produce. We're going to go through yeast cell growth and consumption. We're talking about fusels, esters, phenols, and then some off flavors, mainly being diacetylacetaldehyde and a little bit on sulfur. So a disclaimer again, same as earlier. The goal of this podcast is not to teach scientists. The goal of this podcast is to make you a better brewer. And to do that, we're going over the basics of this. So we are going to be introducing a large amount of topics that we will dive into at later podcasts or crowdcasts going forward. But today is just about laying the basics. It's about making sure that everyone understands the basic principles so we can then delve into these in deeper content later on. Now, as Richard mentioned, if you do have questions, please post them into Crowdcast. We can answer them either at the end or in our large Q&A session we're going to be doing as the fourth lecture in this series. But today, we'll talk about how yeast cells produce flavors and what we taste and why they produce these flavors. That's the big portion. How and why yeast cells produce these different compounds. Not just how to control them, but actually understand why the yeast cells are doing this. Now, we're then going to finish this off in part 3 as to how we can control them, how you can push them one way or the other. Today, we're giving you guys, we're identifying what the levers are. Next time, we're going to teach you how to use those levers to best optimize your brews. That's just how we're progressing amongst all 3 of these. Now, keep in mind with our principles, we need to ask the most important question, and this is by far the most important question. And it kind of emphasize what we talked about last week. Why do yeast cells make flavor compounds? This is probably something that most of you have never asked yourselves. We all just know that yeast cells produce flavor compounds, but never really asked why. Now, the answer here, and this is the important thing to emphasize, is it's not for our benefit. We take the things that yeast cells do and twist them and manipulate them into our own, and for our own well-being. But they do produce flavor compounds for their own benefit. So that's how we're going to be viewing a lot of things we look through today. Why are they actually doing this? Now, flavor compounds are produced to aid the yeast cells themselves, not us. Yeast cells don't care about us. They're just there to aid themselves, which is fair. Now, humans, as we try to manipulate what the environment and the cell health and all the different things we can potentially add into these yeast cells in order to coax them to produce the flavors we want. But at the end of the day, they really are producing these things for themselves, for their own benefit. And that's what we'll be focusing on. Today is kind of best looked at like this: How to Win Friends and Influence Yeast. It's bestseller. Nate, I could be the author of it, but this really would be the book for today. Understanding how yeast actually works and the reasons they're doing the things they are is the first step to understanding how we can manipulate them. And that's what I hope to accomplish today. Now, we're going to break these different flavor groups, or these different classifications of molecules, into 4 main groups. And those are Fusil alcohols, esters, phenols, and then off flavors. Off flavors might seem like a very large topic for us to tackle. However, a lot of the things that we see in fusels and esters and phenols and other things like that, off flavors are very similar to those. So we can kind of cluster them in together at the end. Now, before we go into any of this, there is something we have to talk about first, because this is going to form the foundation in which everything else we talk about today is built on. And that is, we have to talk about how the cell actually works, how the cell actually functions, how the cell grows, how yeast cells grow. Because without understanding that, we can't understand what the yeast cell is trying to achieve. And all these flavor compounds we're looking at, all of them that we're working with, are all a result or related to, some way, shape, or form, how the cell grows. Now, when it comes to flavor compounds function, we're talking, why the yeast cells produce it, not what flavor compounds we get. They have one of 2 main purposes. Now, they're either first to remove something toxic, which we'll talk about heavily today, or secondly, they are a byproduct of some sort of essential process. They are, the offcuts if you're trying to do any sort of construction. They are the things that are just produced as you are trying to construct something that is required. Now, in order for us to understand where all these offcuts are being produced or where these toxic compounds can come up, we have to look at cell growth and basic metabolism, which is where we're going to be starting our lecture today. So no matter what strain you're working with, all brewers' yeast follows the same basic steps when it comes to growth. And to a lesser extent, almost all yeast follow this. So to a basic degree, they all have the same basic steps. Now, unlike humans, yeast cells reproduce via mitosis, which means that the new cell that's produced is a completely equivalent, completely homologous DNA, the same genetics from the mother cell to the daughter cell. So when yeast cells are growing and dividing, that initial large mother cell that produces the new cell is going to just slowly produce a smaller and smaller and smaller, larger, growing daughter cell, which will then bud off and do the same thing as the mother cell did. Now, before we go into the next slide, I want to remind you of this. I'm only going to be showing you information that's going to make you a better brewer. So if I show you something, I think it's important that you need to be aware of it or know it to a lesser degree. We need to talk about cell replication. So we're going to do that right now. Now, cells grow in roughly 4 unique steps. Now, each one of these steps has certain check marks, certain things that occur in each step, and certain thresholds that are required to meet before it's allowed to pass to the next step. Now, if requirements aren't met due to, say, a lack of nutrients when the yeast cells are growing during a fermentation, they won't progress into the next step. The cell won't stress out, and it will just stay in that current stage. Now, as we have a fermentation occurring, we're going to see the yeast cells go through these stages, around and around this cycle, going from the G1 phase, S phase, G2 phase, M phase, around and around and around this circle. Typically in a fermentation, we will see about a quadrupling the amount of cells. So if I added 100 billion cells in, I'm likely going to get 400 billion cells out. Now, this is strain dependent, environment dependent, variable dependent, a whole sorts of different sorts of things go into this. Which is not the point of today, but I don't want to say that it is very multivariable. Now, to give you a bit more of an idea as to what each one of these stages does, the G1 phase is essentially just cell growth. Everything keeps on growing. This is essentially akin to a small child becoming a teenager. Now, in the S phase, the DNA synthesis starts occurring. So the yeast cell will take its 2 sets of chromosomes and duplicate it, so it now has 4 sets. We'll also start seeing the bud formation occur on the cell, as you can see on the side. On the right-hand side. During the G2 phase, we see growth continue. We see continued growth, but more importantly, we see certain core organelles, certain core functional pieces of the cell start trafficking themselves inside of that new bud scar, or that new bud. And then during the M phase, the chromosomes fully divide, and we see the yeast cells separate into a new mother and daughter cell. And pictographically, we can see it occur right here. So on the left, we start off with a cell that would begin its G phase. So the yeast cell gets larger and larger and larger. And then right around the third image in, we start seeing the yeast cell form a bud scar. Sorry, form a new daughter cell. It starts transitioning certain pieces of information, the chromosomes or organelles, mitochondria, things like that, into that new daughter cell. And then once we have full health and full cell chromosomal division, which you see on the second last image to the right, we now start to produce our own unique new daughter cell. And again, this daughter cell is completely genetically homogeneous to the original cell. This is why we don't see large amounts of variation of flavor as yeast cells grow. If we didn't see this, yeast cell population's flavor would shift all over the place. Now, almost everything we do in a fermentation or when it comes to yeast is governed by this process. There's one other thing I want to point out here. So there's one other step for this cycle we missed, and that is what we call the gene knot phase. Now, we've all gone through fermentation or performed maintained fermentations in our households or commercially at workplaces and things like that. And what we see happen in this wheel, in this phasing wheel, is when the yeast cells are dormant. So after the end of a fermentation, or when we've purchased them in, say, a yeast pack or a dry yeast or whatever source you get them from, those yeast cells are actually in this G0 phase. The G0 phase is dormant. And some strains have a really hard time getting out of it. Some strains do so with no issue, no cares. This is part of the reason why we see variations in lag phase. Now, we're going to talk about this a lot more next week when it comes to cell managing and alterations and how we keep yeast cells properly healthy and things like that. All we're trying to do there is figure out how we can reduce the transition from this, the 4 different stages of cell growth and this G0 phase. So the next thing we need to talk about is how yeast cells consume their sugar or consume their food. Yeast cells do consume their food in a very certain way. It's a very consistent way, it's a very logical way. They do so in the following order. They go glucose, arguably sucrose, which we'll see in the next slide, fructose, maltose, and then maltotriose. Now, the reason they do this is very simple. Glucose and fructose and sucrose are some of the smallest sugars they can work with. They're absolutely the smallest. They require, and this is the important bit, no prior degradation for these sugars to go into the rest of their metabolism. Now, if you akin this to a human, when we eat food, very complex carbohydrates or proteins or things like that, the first thing it does is it hits our stomach, which has large amounts of stomach acid. Now, that acid helps break down all this food. It helps break, these really large carbohydrates and substances into very small, easy to manage, easy to absorb, easy to process foods. Yeast cells have to do the similar thing. So the simplest, but it's easier for them to eat foods that do not require any of this pre-processing. Same as us. It is easier for our bodies to absorb things like sugary soft drinks or just pure table sugar. It's much easier for our bodies to consume things like that than it is for us to, say, eat whole grain breads or oats or things like that. It's much more difficult, and it's a longer lasting carbohydrate because of that. The more energy the yeast cell has to expend in order to break this sugar down, the later in the process it's going to do so. And we can see that exactly right here in this chart. So what you see here is the first sugar for yeast cells to consume is glucose. Now, the reason for this will be quite clear in a few slides, but glucose is the first sugar compound that goes into what we call glycolysis or sugar degradation. We'll talk about that in a few slides. Glucose is the first thing that goes into that process, followed then by fructose, which you can see right here, which is why the second thing that goes down. Sucrose does a weird thing down here at the bottom. This is because sucrose is first what we call inverted. Sucrose is made up of one molecule of glucose and one molecule of fructose, which is why you see the fructose concentration increase. This fructose concentration increase is due to sucrose being broken down, the glucose being consumed, but the fructose being preserved. Once all the glucose is complete, you can start seeing right below it, it starts consuming maltose. Now, maltose is just 2 molecules of glucose attached. It's glucose, glucose. That's it. It's harder to break down. We have to break that bond between the two apart. The yeast cell has to spend energy in order for that to occur. So the yeast cell is going to do so second. And then once all the maltose starts to be depleted, what we start seeing then is the maltotriose. Now, maltotriose is glucose, glucose, glucose. It's 3 molecules of glucose. It requires 2 bonds to be broken in order for any of that energy to be accessed, which means the yeast cell has to spend much more energy towards breaking this sugar down. We'll talk about this actually outside of this webinar series, but there are ways in the mash that we can actually alter the breakdown of these 4 different sugars. If we do certain things in the mash, we can get more maltose or less maltose, more maltotriose, less maltotriose, and that will alter fermentability and the final dryness or sweetness of the beer. That's a talk for a later day. As I mentioned before, all these sugars enter the process that we call glycolysis. And as an aside, humans do have exactly the same thing. This is the first step for humans to consume sugar as well. Almost every single eukaryotic organism on the earth, this is how it starts. I don't know of one that actually doesn't start like this. Now, the goal of this process mainly is to produce energy. Now, the cell does this in 2 main forms. There's 2 main energy currencies we're looking to produce. And the first one, which is the one that most people have heard of before, is what we call ATP. Now, ATP, you've probably heard associated with the mitochondria being the powerhouse of the cell. And ATP is by far the general purpose energy for the entire cell. It is used everywhere to do all sorts of different things. But another important one that's very important when it comes to brewing is what we call NADH. And this is a cofactor that provides energy for reactions to occur within the cell. And what we're going to see throughout today is that if we're deficient of NADH, we start having problems when it comes to off flavors or incorrect flavors being produced. But we're going to save that for a few slides from now. We need both in order to be functional, not just one. Now, this is the complicated form of what glycolysis is. Now, again, I don't expect anyone to be able to recite this. It's not going to make you a better brewer. Being able to draw the whole thing out, it's going to be a cool, maybe not a cool party trick, but a party trick nonetheless. I had to do this during university, and some of you may have seen this and just kind of shuddered a little bit from some flashbacks there. What I would recommend you focus on is this chart right here. And this really, from a brewer's standpoint, is all the information you have to know. That is the following. Glucose, as I see on the left-hand side, comes inside the cell. And through the glucose being broken down, we generate several things. We generate 2 molecules of ATP, 2 molecules of NADH, and 2 molecules of pyruvate. That's it. That is all you need to know when it comes to glycolysis as a brewer. Glucose goes in, 2 ATP, 2 NADH, 2 pyruvate. Now, this is incredibly useful. Glucose has come in. We now have produced a large amount of energy, which we can now use to spend on all the different things that are required to do inside of the cell. And this is how the yeast cell generates effectively all of the energy that it uses to function. We're going to pick this up a bit later as we see how this sugar starts going different places, and more importantly, how that pyruvate on the right-hand side starts being broken down. Or we see it right here. We mentioned last week about the differences between aerobic and anaerobic respiration, showing that aerobic respiration produces roughly 36 ATP, while anaerobic fermentation produces only 2. 18-fold increase in energy, which is massive. This is part of the reason why oxygen becomes so important. Now, when we give the yeast cells oxygen, this energy boost allows them to build up reserves. They don't just use the stuff right away. They're able to actually accumulate this for later on. And that's incredibly important. It's incredibly important because this allows the yeast cell to harbor or to struggle through more stress. It's able to go through the bad times. It's able to push through something that's difficult and keep on surviving. If we have cells that have not gone through a large amount of aerobic respiration or haven't seen oxygen in a very long time, and they're hit with even the smallest amount of stress, the chance of those cells being unaffected is very low, very little. We need to make sure that these yeast cells have proper levels of energy. And that's going to be a common theme throughout today. Making sure the yeast cells have high levels of energy so they're able to actually perform and clean up certain compounds we want them to is core and critical for the yeast cell's proper fermentation flavor. A few other things we need to mention. What we see here is a standard graph of gravity, which would be specific gravity. So if we're looking at degrees Plato or SG, temperature, which is held stable across the entire line. This is a logger, by the way, just so we're all aware. That's why it's lower fermentation temperature. And what you see inversely correlated to the gravity is biomass. And this is yeast cell biomass. And this is something I mentioned earlier, but it's really important for us to be aware of. As yeast cells are consuming sugar, not all that sugar is going towards ethanol. A large portion of it is just going towards the yeast cells producing more of themselves. It's going towards biomass. Now, as we mentioned last week, there were two main things that actually go towards feeding yeast cells, actually are used for the yeast cells to consume and respire and produce more of themselves. And we've talked about one of those already. We've talked about sugar. We've covered the close notes of it. But the other one we haven't talked about is FAN. Now, FAN, like sugar, does have a sequence in which it's consumed. Much like how sugar goes from glucose, fructose, sucrose, maltose, maltotriose, FAN does have a similar hierarchy. However, there's a few issues with this. It is poorly understood. It's very strain dependent. And more importantly than that, it typically requires about a 4-year undergrad in biochemistry to understand, or at least something in the sciences. Now, a quick summary for you guys to kind of take away, though. Your maltsters have this information. Almost all malt that's used to produce barley malt has been selected so that it contains a high FAN content useful for brewers. So it has a higher concentration of things that are similar to glucose and not maltotriose. Other things, proteases that can be activated in the mash can help you increase the amount of FAN available, which gives your yeast cells more food and also changes the type of food they have access to. And as I mentioned earlier, in order to really get into this topic, it requires a lot more information, which probably is outside the scope of these lectures. But there are a few things we can take away from this. Now, this is the same data set we saw before, the same time frame, the same experiments, the same fermentations. But what I want to show here is that as the fermentation, as ethanol goes up, which is inversely correlated to the amount of gravity going down, we also see FAN decrease and level off. And this makes lots of sense. This is what the yeast cells are actually using to consume and make more of themselves. They need this. This is the protein that they have to work with. As we see one go up, biomass goes up, FAN has to go down because this is how they actually build themselves up. And if you want to layer the two on top of each other, you can see it right here. Biomass goes up, FAN goes down. Gravity goes down, ethanol goes up. All makes sense. Yeast cells are consuming the sugar in their environment and the FAN to make more of themselves. It's as simple as that. Now, we have more to talk about with FAN, and honestly, I'm a big fan of FAN. FAN is going to be something we're talking about all throughout today because FAN has a huge impact and an often overlooked impact when it comes to flavor compound production. FAN is associated with Fusil, with esters, with diacetyl, with acetaldehyde, with all sorts of different off flavors. Unfortunately, this isn't a scenario where more is more or less is less. Too much can actually be bad. Too little is also bad. So what we're going to be doing a lot of today is exploring each one of the different compounds that can be produced and how it's related to this, how we should try to modulate this and layer this in, because it has a really big impact on your beers. Now, as a guiding principle towards almost everything in life, output is based on input. So depending on the type of FAN, depending on the type of sugars, depending on the type of yeast cells, we're going to be generating different things on the output. Now, I can take the same yeast and I can give it 3 different types of FAN, and I'm probably going to get 3 different types of flavor profiles out. They're not going to be radically different, but they are going to be unique to each other. One's going to be a bit more estery, one a little bit more of something else. The type of FAN, the quality of FAN does have a big impact. So we're going to start going into the next section, which is Fusil alcohols. Now, Fusil alcohols are just bigger, larger alcohols. They're bigger relative to the alcohol that you typically see associated with fermentation, being ethanol, which is 2 carbons. Fusil alcohols are always larger than 2 carbons, which is why they are often called higher alcohols, larger alcohols. Now, Fusil alcohols are almost always seen as a negative in beer. I myself am someone who appears to be a bit more sensitive to Fusil alcohols than most, typically with migraines and things like that, or headaches, which is not great. And the Fusil alcohols are often described as either hot, cheap spirits, headache-inducing, not typically things you want in a beer. At best, they're considered to be just boozy, at best. Now, they typically taste negative, and there's a reason for this. It's actually very hard for our bodies to break these things down. It's also very hard for yeast cells to break these things down. It's not just restricted to humans. It's all organisms. Now, Fusil alcohols are our biggest issue in the bioethanol field, so you're starting to see a lot more research come out from this. And it turns out that Fusil alcohols are about 18 times as toxic to yeast cells as ethanol, as the main thing it produces for all of its fermentation. These things are nasty. They're not easy to be handled with, not for us, not for the yeast. So the question logically comes up, why do they make them? The reason here is necessity. They're a byproduct of an essential reaction. That's what we're going to talk about. Now, what we see here is a standard kind of metabolism, metabolic pathways for a few different common beer flavors. But what we're going to focus here on just the Fusil alcohols, just here in the bottom right. Now, there are two main ways we can see Fusil alcohols be produced. Now, the first way is what we call the anabolic route or the creation route. And this is sugar goes to pyruvate, that thing we just saw at the end of glycolysis, which then goes into some molecule called keto-oxy acids, which then will turn into a Fusil alcohol. We'll talk a bit more at a later point as to why the yeast cells do this. Now, the other way, and the most common way you see in brewing, the way in which these cells produce Fusils, is what we call, or it's called the catabolic route. And this has to do with amino acids, which are a part of FAN, being broken down by the yeast cell. The amino acid comes in, gets stripped and turns into a ketoxo acid, and then turns finally into a Fusil alcohol. The reason the yeast cells do this is they need things from those amino acids. They need things from these compounds. What they really need is they need the nitrogen. And we talked about this earlier this week, where we said that nitrogen FAN is a type of nitrogen molecule. Sometimes the yeast cells need the more purified version. They need a part of a piece of another source of FAN. They don't need the whole thing. They need a certain type. And that's about as in-depth as we're going to talk about when it comes to certain types of amino acids in regards to FAN, because we start getting really into the weeds if we go any further. But the important thing here to note is this: if we do not provide enough FAN or not enough of the right types of FAN, we'll start seeing that anabolic route being produced. We'll start seeing large amounts of Fusil alcohols being produced because we underfed or starved these yeast cells of this core thing it needs. It's going to try and use pyruvate and use its sugars to produce these things that it really should have been given to it in the first place. Now, if we feed it too much, however, we're going to see the same thing occur. We're going to see the catabolic route activate, and we're going to see Fusil alcohols galore. Balance here is key. We need the proper level of FAN, not too much FAN, not too little FAN. We need a proper amount. Now, if you're making standard 1040 to 1060 or 10 degrees to, what is that, about 14 degrees Plato beers, you're fine. You're probably fine, as long as your yeast cells are in proper health and they're doing well. There are some areas, however, when we start seeing issues with either of these two. We're going to explore them now a little bit. But to give you a little bit of a walkthrough, a bit more in depth, what we see here is just kind of the metabolism associated with producing a Fusil alcohol. So on the left-hand side, you see two main amino acids, threonine and leucine. There's about 20 or so that the yeast cells commonly, and humans, require for operation. And you see here is that as the yeast cells try to strip molecules off, they strip the nitrogen off, they strip other portions off these molecules they need for other reactions. The end product, as we move from left to right, is this Fusil alcohol. It's not great. We don't like it. But the end product here is from threonine propanol, which is a common Fusil alcohol. And from leucine, we get isoamyl alcohol, which is common in a lot of Hefeweizens and things like that. Not ideal. So what I want us to do is look through two different examples. The first one here is kind of a high ABV runoff. So the two beers we're going to look at is a Belgian Golden Strong, a BGS. Now, Belgian Golden Strongs use about 20% simple sugar in their grain bills. That's it. It's a very high glucose content beer. Lots of simple sugars. More importantly, less FAN. Because a large amount of the fermentabilities of this beer is coming from sugars and not from barley malt, and barley malt is what contains the FAN, this is going to be a lower FAN beer. Now, to go ahead of that, we have a barley wine or imperial stout, two beer styles that are almost exclusively 100% malt, 100% FAN, very high FAN content. And if you taste these, and even if you look at the BJCP standards and things like that, Belgian Golden Strong should never have Fusil alcohol character to them, while barley wines and imperial stouts can have a hot, boozy characteristic to it. The reason here is simple. Less FAN equals less Fusils. Belgian Golden Strong has a lower FAN content. There it's going to produce less Fusil alcohols via the catabolic pathway. Belgian barley wines and imperial stouts have more FAN. Therefore, they're going to produce more Fusils. We can break it down as simple as that. Now, the second example I want to show you is for light lagers. So if you ever notice that almost all light lagers have almost no Fusil character to them. They can have other problems to them, which we can talk about in the Q&A afterward. But the main reason here is because these beers lack FAN quite heavily. And the reason for this is that corn has very little FAN content to it, much less than barley malt, significantly less than barley malt. So what we see here is less Fusils equaling less FAN, less in, less out. Now, as long as the cells stay healthy, this should be fine. However, If we have unhealthy cells or stress cells, or cells that just, they're not doing their best, something's happened to them, and we give them a low amount of fan, we will get fusels via that anaerobic pathway. It's not ideal. It's something that you typically see large-scale breweries deal with by pushing these beers towards discount brands. Not something you see common in most microbreweries or home brewers, but it's something you need to be aware of. And just to show you guys the data set on corn content, or maize, what we see here is the decreased nitrogen content, which fan is a portion of nitrogen, as we increase the concentration of corn. So as we see, as the concentration of corn increases, the overall nitrogen concentration of the swort starts decreasing in relation to the corn percentage. You don't want to go too high. You don't want to go too high. You can do it 1 or 2 times with the same yeast strain, then it's going to start getting very mad and start producing things you don't want it to produce. So a quick summary on fusels. There are 2 main things to consider when it comes to controlling your fusel levels. Fan levels are the big one. You want to make sure you have the proper level of fan. If you have too much fan, you're going to get too much fusels, or you're going to have noticeable fusels. If you have too little fan and your yeast cells are perfect health, you're going to have less fusels. However, if you have very little fan and your yeast cells are stressed, you're going to have more fusels due to that anabolic pathway. Now, you also want to keep your cells healthy. Stressed cells typically require more fan. They have more things to repair. They have more things to build. They require more building blocks. Healthies consume less fan. They don't have anything to repair. They're good to go. They have nutrient reserves. They're fine. They require less fan, therefore less fusels. So with that, we're now going to switch gears into esters. Now, esters are almost always seen as a positive thing in beer, and for the most part, that is correct. If you really push ester concentration high, you can sometimes get this kind of nail polish character, but that's very rare and kind of few and far between. Esters are often described as fruity, tropical, or candy-like, and the type of esters you get are often very strain-dependent. So to kind of keep things consistent, we have to ask the question, why do yeast cells make them? The answer is 2 reasons, actually. Well, first is for proliferation, and the second one is for detoxification. So we're going to break these 2 down. So proliferation is, esters are commonly flavors that are found in nature. A lot of flowers and fruits contain esters. Now, they taste positive to us because they are found in nature in these things that our body deems flavorful. And not just us, deers, insects, it's the big one. Yeast have mimicked these flavor compounds from other organisms to help them move around. What I mean by this is the following. So an experiment was done a handful of years ago where they took yeast cells and they knocked out the ability for them to produce esters. So these are ester-negative yeast cells. And the interesting thing was that when you took fruit flies, other insects, they showed no interest in these yeast cells that were devoid of esters. They didn't smell good. There's no reason for the flies to go there and get that thing thinking it's food. However, when you have yeast cells that are able to produce esters, they start flocking to the yeast cells that are producing these flavor compounds. And this is important. Fly is one of the main ways in which you see yeast cells travel across the world. Yeast cells don't have legs. Yeast cells are completely dependent on other things to travel around from source A to source B. There's no way around it. So making sure the yeast cells can attract flies is incredibly, incredibly important. Now that's the first part. That's proliferation. The other part is detoxification. Now esters, as we showed before, fusels are much more toxic than ethanol for humans and yeast cells alike. Now the process of esterification allows us to break these fusels down into more positive esters, less toxic compounds, and that's the big thing here. The process of esterification is just an acid binding to an alcohol, those 2 fuse, turning into an ester. Now when it comes to yeast cells, this esterification occurs via a few different standard molecules. We have acids, mainly being acetyl-CoA, which is found inside the cells, and then we have an alcohol. In this case, it's almost always the fusel alcohol. The thing that's toxic, the thing that the yeast cell wants to break down and remove from its environment because it's harmful. These 2 bind, and the byproduct of this is something that smells very fragrant, very lovely, attracts flies, and makes us want to consume it. It's a wonderful scenario, wonderful system. So the simplified version for this, or for how we get different kind of esters and different flavor compounds and things like that, is quite simple. Different yeast strains require different amounts of fan. They require different amounts of certain amino acids. And based on the amino acids they consume, based on the types of fan they break down, we're going to see different Fusil alcohols be produced. Now, if I have, say, 6 different Fusil alcohols and I esterify them all the same way, I'm going to get 6 different types of flavor compounds that all taste different. And that's the big part of the reason why we see large amounts of variation in the types of esters we see from, say, an English strain to an American strain to a New England-style IPA strain to a Belgian Trappist strain. It all has to do with the type of Fusil alcohols they are producing based on the fan they consume. The way to look at this, the more Fusils we have, the more potential, and this is the important bit here, the more potential esters that are being produced. So the question now becomes, how do we take these Fusils that are present inside of our beer and help ensure that they're actually going to turn into esters on the other end? So esters are a complicated topic, an incredibly complicated topic, one of which that we'll actually be talking about in later webinars, in later series. However, there is a Cole's Notes version that we can talk about, and that is the more oxygen, the more esters. If you want to get more esters, add more oxygen. If you want to get less esters, decrease oxygen. Now, a handful of you are probably thinking, this is what I see with my students all the time. They'll go like, I knew this, I knew that, but I don't know why I know that. And the answer here is actually old school. It's incredibly classic. And I want us to take a quick look at different types of fermenters, different types of ways in which fermentations were managed and outside of the modern era. So what we have here is a brewery of the open fermentations vessels at Schneiderweiss. And what you're seeing here is the yeast cells go to the top of the fermentation. It's a top cropping Hefeweizen strain, as almost all Hefeweizens are. What's happening here is that as these yeast cells go to the top, they are accumulating a bit more oxygen during this fermentation. When you have an open ferment, more oxygen is able to get inside the yeast cells, which means that they're able to more effectively turn any Fusils that were being produced into esters. Now, they're not alone. It's not just the Belgian brewers, the Hefeweizen brewers who are doing this. If we look at Sierra Nevada Brewing, for their Narwhal Imperial Stout, for their Bigfoot Barley Wine, for a lot of their very large, high ABV beers, they only ferment in these large open fermenters. And the reason for it is the same. They want to try and maximize ester profile. They want to make sure that any Fusils that are being produced during the fermentation, they want to give them a little bit more oxygen, hopefully turn those into as much esters as possible. A little bit of oxygen during fermentation, less Fusils, more esters. They're not alone. This is the old Burton-on-Trent. I can't recall the exact name of the system for it, but this is an old school way in which English-style brewers would ferment. Essentially, there's a whole series of barrels underneath here, underneath all these different tubs. As the barrels start to ferment, they start overflowing into the tubs, aerating the wort during fermentation, allowing for more esters to be produced. In Ontario here, there is a local brewery that aerates, in Toronto, there's a brewery that aerates midway through fermentation to try and get more esters and make their yeast cell more happy. And it's not just exclusive to ale brewers. If you look at Pilsner Urquell, which you see right here, using some of their old school wooden barrels, they ferment open. Any Fusils that are being produced during this ferment, you're going to have a little bit of oxygen come in, and they're going to be turned into esters, giving you a more flavorful, vibrant beer. This is old school. This is kind of common knowledge, but something that during the modern kind of homebrew and professional brewing ethos, we kind of lost a little bit. So as some general rules, if you want to increase your ester profile, open fermentation, more oxygen, you're going to get more esters. A conical fermentation, where you have a very small 1.5-inch, often TC, triclant port for all your off gases, you're limiting the amount of oxygen that your yeast cells have access to, you're going to get less esters. A carboy fermentation, it's all glass, oxygen can't permeate through, you're going to get less esters. If you do a yeast starter where you're bombarding those yeast cells during the ferment, during their growth with tons of oxygen, they build up oxygen reserves, and you're going to get typically more esters. If you have old yeast cells that have been sitting around for a while, that depleted all their oxygen reserves, they haven't seen oxygen in a long time, you're going to get much less esters. If I use fresh yeast, things that have a high amount of intracellular oxygen reserves, I'm going to get more esters. If I have freshly harvested yeast cells and I give them lots of O2 when I knock them out into the next beer, I'm going to get more esters. Now, there are a few other things for this. Fermentation temperature, pitch rate, those are absolutely things that are going to impact the amount of esters we produce. But that's what we'll be talking about next week. And with that, we're going to move on to to phenols. Now, phenols are arguably one of the easiest flavor compounds to control. Now, when desired, phenols are incredibly beneficial. They're incredibly, if you're looking for Hefeweizen or a wit or a saison, you want phenols. You want that nice spice characteristic. It's the thing that actually separates it from other styles of beer. Now, phenols, like esters, are produced due to the breakdown of a toxic compound. They're trying to turn this toxic thing into something that is less toxic. And most phenols are a result of ferulic acid being broken down. And they turn it into 4-VG, or 4-vinyl glycol, the clovey characteristic we see inside of a lot of these saisons, wits, and things, and other beer styles of the ilk. Now, ferulic acid is mainly found in wheats, oats, spelts, kind of all things that aren't barley. Now, barley does contain a good amount of it. It's just not a high enough concentration to really stress these cells out. It's why whenever we look at beer styles that have that kind of clovey characteristic, a wit, large amounts of wheat, or even oats, saisons, good amount of wheat, often. Hefeweizens, up to 60% malted wheat, even higher in some cases. They're all beer styles that contain a high concentration of ferulic acid, the precursor to turn these, that needs to be present to be, the precursor to turn into 4-VG, that clovey characteristic we see. Now, a few of you are probably thinking, but wait, I've seen variations in this. I know this isn't the only thing. And this is the important bit here. Often people will say that, there's different types of phenols, or, sometimes the clove comes out as just pure clove. Sorry, the 4-VG comes out as pure clove, or sometimes it's more of a baking spice, or sometimes it's more complex. What you're often seeing when it comes to these things is you're seeing a few different variations, but not in the phenols itself. It's often associated with the flavor compounds that are around the phenols. To give you an easy example for this, if we take a Hefeweizen and we ferment one very warm, we're going to be driving large amounts of esters, high amounts of esters. So we're going to have a high amount of esters and a stable level of clove. If I ferment the same beer at a lower temperature, my ester content will go all the way down, but my clove stays relatively the same. The clove content is relatively unimpacted, so to speak, by the fermentation temperature, other variables. So in that first scenario, I'm going to taste a lot less clove because there's all these other compounds on top that are crowding it out. However, if I ferment cold, that clove will be much more prominent. And what we see when we have this, the esters and other compounds will actually influence how we perceive the clove, changing it, either its intensity or what flavors we actually taste it as. All we're going to talk about for clove, because we have a few other things I want us to really hammer on. Now we're going to move into off flavors. Off flavors are a very large term for a handful of different types of compounds that are out there. However, for today's focus, we're looking at the two main ones we see in beer. The first one we're going to talk about is diacetyl. Now, diacetyl, hang on. There we are. Diacetyl, much like fusels, diacetyl is a byproduct of yeast needing to produce an amino acid. In this case, it's valine, where fusel alcohol is the anabolic pathway, whereas they're trying to take sugar and turn it into this into this amino acid. Diacetyl is the same thing, but diacetyl is a byproduct of it needing that valine, needing that amino acid it's trying to produce. Now, there's a few differences here. The big one here is that with fusel alcohols, we can't get rid of them. We can't get rid of fusel alcohols unless we esterify it. It's really the only way. And if we don't esterify it, it's locked in. We're stuck. But diacetyl, we can get rid of it. Now, diacetyl is naturally reuptaken at the end of fermentation. However, trying to determine when it's actually completed can be tricky. And the reason, and we're going to talk about this right now as to why this is, but we have a solution for this. It's something called the forced diacetyl test. Now, I just want to point this out. We've already seen diacetyl on this sheet. We've already kind of walked past it. And that's in the form of vicinal diketones. That's another name for diacetyl. Now, here's the full metabolism we can look at when it comes to diacetyl. Now, glucose in this case goes to glucose to pyruvate, and then pyruvate to a molecule we call alpha-acetylactate. This is important because alpha-acetylactate is the main precursor for valine, one of the amino acids the yeast cell needs to grow. Now, we can bypass all of this if we just give the yeast cell valine. If we give the yeast cell amino acids that contain, or FAN, that contains this amino acid. It doesn't have to do it. It's wonderful. We supply the proper levels of FAN and those proper portions. They don't have to go through this entire pathway. It's wonderful. However, if we find ourselves in this situation, which we often do, the yeast cell has to produce more valine. In this case, we'll see glucose go to alpha-acetylactate. And what ends up happening is that not all that alpha-acetylactate is turned into valine. Some secretes out. It leaves the cell. And the important thing here for us to note is that this alpha-acetylactate is odorless, tasteless, effectively not able to be sensed by the human palate. However, if we give it a little bit of oxygen and we increase the temperatures, such as, say, a quick packaging run and then the beer being sitting on the LCBO shelf for a while, or whatever your local alcohol distributor is, this will cause this alpha-acetylactate to break down into diacetyl. So I'm sure we've all been in this position where you've bought a beer from some sort of distributor of beer, and you can bring it home, you put it in your fridge, you open it up, you pour it, you try it, and it's just a horrific butter bomb, diacetyl being the main thing that tastes like butter. And you think to yourself, how in the world could this brewery release this beer to the public? This is butter. Did the brewery not notice this was butter? And what you've actually seen here is this brewery tasted their beer in-house, tasted fine, didn't have a single problem to it. But the beer had a high concentration of alpha-acetylactate. They package it, it picks up a little bit of oxygen, it sits on a store shelf for a while, gets warm, high temperature, the perfect storm for alpha-acetylactate to be broken down into diacetyl. Now this is the next catch. Diacetyl is only able to be absorbed by the cell. The cell can't reabsorb alpha-acetylactate. It's not able to do so. It needs the alpha-acetylactate to be broken down into diacetyl. So once we see all that broken down, then the yeast cell can bring it inside the cell, turn into acetone, and then finally 2,3-butadiol. This is important. So if you want to find out if your beer has this alpha-acetylactate problem, it's a pretty easy test. And if everyone's curious, this is Alex. He's wonderfully modeling this for us. All you do is you take a small sample of your beer. It can be a test tube, can be a mason jar. That's what I often use. I've even seen some people use water bottles. Put a small sample of the beer inside that container, and then simply chuck it into a hot water bath. Now, it doesn't have to be fancy. It can be a pot of water that you've recently brought to a boil and then thrown the sample in. That's what I usually do with mason jars. Commercially, it can be a bucket that you fill with water from the HLT and then throw the mason jar in. You forget about it. Come back 30 minutes, 40 minutes, could be even an hour later. All you then do is you open the vessel and you smell. If the sample smells like butter, you actually have alpha-acetylactate inside of that beer. And through taking the sample and heating it up, you've actually broken down very quickly all the alpha-acetylactate into diacetyl. Really easy test to see whether or not your beer is able to be cold crashed or able to be packaged or pushed forward in the process. Very, very useful test, by the way. Now, the next big common off that we're going to talk about is acetaldehyde, which is kind of a pet peeve of mine. It's a flavor resulting in the buildup of a byproduct during fermentation. Now, acetaldehyde is mainly due to the cells being unable to complete fermentation. Now, I mentioned at the top of this that NADH is an important molecule, and this is where it's going to rear its head up again. NADH, the molecule that helps catalyze certain reactions, this is where it comes into play. Now, in order for acetaldehyde to be broken down into ethanol, NADH is required. So if the yeast cell has used that NADH in other means, we're going to be stuck with acetaldehyde. Now, much like we saw with Fusils, acetaldehyde is actually toxic to the yeast cell. The yeast cell really does not like it. It wants to break it down. It's doing so because it can't, or it's not doing so because it can't, because it's run through all these different energy resources. Now, much like we saw with Fusils, if we want to get rid of Fusils, the solution is actually quite similar. More oxygen. We give the cells more oxygen. They get more energy. I always said at the top, they go through aerobic respiration. They produce 36 ATP instead of 2. They are able to maintain and increase reserves of NADH. They're able to handle this. They have more energy. They're going to get less acetaldehyde because they have the proper requirements to break it all down. Acetaldehyde is almost always a byproduct of the yeast cells just not being healthy, of the yeast cells not having the proper nutrition, not having the proper aeration levels, not being managed properly, being stressed out. And to see where acetaldehyde actually comes from, we can look at this chart that we saw earlier this week. So we see the top part here, glucose to pyruvate, that's just glycolysis. That's the standard metabolism we've already talked about. Where we then start seeing the rest of this pathway go is we go from glucose to pyruvate, pyruvate to acetaldehyde. Now, in order for acetaldehyde broken down, we actually have to spend energy. You can see this right here. The NADH that was produced during fermentation all goes to producing that ethanol, all of it. 2-acetaldehyde in, they're used to actually break this acetaldehyde down into the ethanol that we were looking for. The net sum of this pathway is 0 for NADH. We're producing ATP, we're producing energy, so that's good. But we need NADH not just for this process. NADH is used all throughout the cell, and if we don't supply the yeast cell with proper oxygen, they won't have enough of that energy. If we give the yeast cells oxygen here, this pyruvate doesn't go into acetaldehyde, it goes to different pathways. The NADH is allowed to be preserved. If we give it oxygen, actually, the yeast cell via other means starts producing large amounts of NADH. It's much more healthy. If you really want to make sure your yeast cells are healthy, you need to give them oxygen. We'll talk about that more next week. One quick side note here, and this is one of my favorite metabolism graphs you see right here, and this is the reason why. When I was early in my biochemistry career and looking at fermentation, probably I should have been studying for some sort of final, but instead I was looking at beer stuff. I found this and it blew my mind, because if you follow this through, glucose to pyruvate, pyruvate to acetaldehyde, and this CO2 that's produced right here, this is where almost all the CO2 that we see occur and ferment is produced. Almost all the CO2 we see that's used to carbonate our beers occurs right there. Almost all the CO2 we see causing all the rousing and mixing during fermentation is all from right there. It's really cool. We actually can look at this down to the metabolic and down to the molecule and see where it comes from. I just find it absolutely fascinating. Now, one other last thing with acetaldehyde, and this is how you actually taste it. Now, the flavor of acetaldehyde is quite nebulous. When you look at it, and the kind of classic flavor profile of acetaldehyde is that fake green apple. Now, acetaldehyde, I mentioned light lagers earlier. Acetaldehyde is common in light lagers because you're not giving them enough fan. The yeast cell has to produce so much of these amino acids that it has no NADH left, so acetaldehyde accumulates. It's just what happens. And when you have nothing else going on in that beer, and I mean, one of the best backhanded compliments you can give is saying that, yes, Coors Light tastes like nothing. It's incredibly impressive that it tastes like nothing, because so many things can go wrong, but it tastes very close to water. And though in that scenario, yes, it tastes like fake green apple. But when you start putting this into other beers like IPAs or stouts or porters or pale ales or saisons, it does not come across the same way. Oftentimes, and this is almost all flavor compounds, it will couple with whatever's in its environment. So at a low dose, how it's often perceived is there's something off, but I can't describe it. And I'm sure we've all been in this position where I know there's something weird, there's something that's not quite right, but I can't tell you what the flavor compound is. It's usually acetaldehyde. And a medium dose can often come across kind of a rotten fruit. I sometimes get like a freshly cut pumpkin or some sort of gourd, like butternut squash or something like that. What other compounds are surrounding it will change how you actually taste it. But in almost every single case, it's not a good thing. You want your yeast cells to be happy. You want your yeast cells to be healthy. You don't want this in your beer. Now, to bring this all together, I just want to walk us through this a little bit. I showed this chart earlier this week, and I said that we're going to get to this chart. And I know when you first look at this chart, it can be quite overwhelming. And there's a lot of things going on. What I want us to show is that just through the things we've gone through today, less than an hour, we've actually covered most of this. So we started with sugar. We talked about how yeast cells actually consume the sugar in their environment, whether it be glucose, sucrose, fructose, maltose, maltotriose. Break it down, and it goes sugar. Go through like glycolysis, we get pyruvate. Pyruvate can then go to alpha-acetolactate, which can turn to diacetyl and be excreted, which we then get as an off flavor. Alpha-acetolactate can also go to keto acid, start producing valine, which is required for cell growth. That's an amino acid. Now, amino acids can also be brought in from the outside of the cell. These can be broken down into keto acids, aldehydes, and finally higher alcohols, Fusil alcohols. All three of these can be excreted outside the cell, which can be various types of off-flavor compounds. Now, as well, if we give pyruvate, we give it oxygen, it's going to turn to acetyl-CoA, and then once that acetyl-CoA is going to bind with higher alcohols and give us esters. If we don't give it that, we don't give it oxygen, that pyruvate is going to turn into acetaldehyde, which can excrete and leave the cell and give us an off flavor, turn to ethanol, and then leave the cell. We covered most of this just in small little bits. This graph isn't nearly as large or intimidating once you start breaking down its individual components. And this really does summarize all the different things that can go on inside of the fermentation cell from a brewer's standpoint. Now, there is one last thing I want to talk about, and this is sulfur. And we've been talking a lot about amino acids, a lot about different fan compositions today. We're going to talk about a bit more now, but this is the last thing we're going to hit on. Now, sulfur, we've all had this with lager fermentations, or even at the beginning of early signs of a fermentation. You can sometimes get a sulfur aroma from the beer. Now, a lot of people suggest that this is actually an off flavor. This is something that's negative, something that's non-desirable, or an error in a sense. But you're almost always going to get a little bit of sulfur. And the reason for this is twofold. Much like we saw with Fusils, certain sulfur-based amino acids, or certain amino acids require sulfur. They need sulfur in order for their creation. And if the yeast cell is not supplied with those proper amino acids, it's going to have to produce them themselves. And if it does so, you're going to start seeing sulfur compounds start leaving the cell. And that's where a lot of the sulfur compounds we see during fermentation occur. They're actually the yeast cell having to build up amino acids that were not given to them. Now, if we can supply them with the proper amino acids beforehand, much like with Fusil alcohols or even diacetyl, they don't have to make it. If you give the yeast cell the proper amount of fan or a higher level of fan, the chance for off flavor or just flavors you don't want is going to decrease. And in some odd cases, much like we saw with Fusils, where the fan was given and then they needed that nitrogen, they need those components to it. You see the same thing happen with this, where some sulfur compounds can be released as the yeast cells strip off the sulfurs for other portions of the amino acid that was required. Fan is a really big deal. And I hope I've kind of hit that on the head today. So with that, we've covered a lot of content. I hope you guys all kind of enjoyed that kind of walk through basic flavor compound production. I'm sorry about the dog barking and things like that, but I think we have some time for some questions.
All right. Thanks, Nate. A lot of information there. I hope everyone's digesting that now.
The fact that you can record this is going to be a benefit.
Yeah, absolutely. You can go back and look at anything if you want to brush up on any of that knowledge. Before we launch into the questions, I just want to mention that we'll be hosting the next installment of Yeast Basics this coming Monday at 8:30 p.m. And then we'll be talking about harnessing yeast flavors at that point. So sort of taking everything we've learned so far and actually starting to understand, what choices can we make as brewers in our ingredients and in our brewing process to really get those flavors that we're looking for out of the yeast. So I think that will be a little bit more practical, a little bit less, super technical as this one. And we'll really understand how to control the flavors.
One other thing I would like to mention, we went through a lot of content today, and this is because this is the basic foundations of how all these different kind of flavor compounds are produced. I'd recommend that you've section them off if you're going through over this again. Look at Fusils, spend some time with it, and look at Esters, spend some time with it. Don't try to tackle everything as one large chunk. Every single thing has the same underlying principles, but does have its own kind of quirks associated with it.
For sure, yeah, and I think it's important to understand, tie it all back to just like the yeast metabolism, understand that everything's tied together, and there's a reason that all of these flavors are made. Mm-hmm. I also want to mention that once we're done with this Yeast Basics series, we're also scheduling some new webinars. On April 6th, we're going to be talking about yeast strain diversity, some of the research that we've been doing on our own yeast strains and explaining some of the differences in flavors and in other things like nutrient consumption, all of these things that really impact how those yeast behave and how they're handled by the brewer. And then on April 9th, we're also going to take a deep dive into biotransformation and walk through that process. I know tons of people are making hoppy beers, and this word gets thrown around, and we want to explain what it means and also how you can actually harness it as a brewer. So we're going to launch those sessions for signups shortly. And
all those things will be building on what we covered today. They'll all be taking kind of these basic principles and pushing them even further.
Oh, yeah, it's all connected.
There'll be some review. Don't worry. There'll be some review.
Of course. We've got some time for questions. So I think we'll just like last time, we'll go down the list.
Sure.
We'll go just in order of the upvotes. But of course, I just want to mention that we are recording every question that we don't answer. We're recording it in a document, and we're going to try to put together a list of questions and answers for our Q&A session. And there's a lot of common themes that are coming up. So we'll try to address those questions as efficiently as we can and try to pass along a little bit more knowledge.
Yeah, with a bit more information, it won't just be us talking. We'll have slides and things that are presented for the question. So if you mention a question, sometimes it might just be that's too complex. We have to answer that in slide format. We can't answer that just talking. Yeah,
there was a great question last week about the impact of zinc on flocculation, and we didn't really know the answer, but we're starting to dive into that and finding some really cool stuff. So we're learning as well.
Yeah, let us know what you want to know.
Totally. So Nate, I've got the first question for you, and this is really, honestly, one of the most common questions that we get from professional brewers, and honestly, still a hard question to answer. So the question is, when you're doing a multiple batch brew and you're filling a tank over 2 days, should you be oxygenating your batch on day 2 if the batch is already fermenting?
Oh, I want to give a whole lecture on this. That's something exactly like what we just went through, but just on this topic.
This is something we've talked about, actually, is having a whole webinar on oxygen.
Yeah. The gut reaction is no, and in rare cases it's yes. So if you're making something like an English style ale or a Trappist style ale, then it's pretty common in those scenarios for not necessarily aeration to be applied, but for oxygen to slowly egress during the fermentation. So in which case you can aerate, but it can cause some problems. And this is the important thing to kind of make sure I highlight. If you put yourself in the mindset of a yeast, yeast cells kind of have two streams. We have that aerobic respiration, we have the anaerobic respiration. And some yeast cells have no problem switching lanes, no problems. Other ones are really bad at it, to the point where if they're going through fermentation and then all of a sudden you give them some oxygens, they flip. And then they try to go back, but they can't quite go back, and the cells just go into chaos. They run through all the nutrients. It's not good. Other cells have no problems with it. The English strains and kind of the top cropping strains don't seem to have a big of an impact for it. If you want to go with more so the old school kind of clean ale, clean lager methodology, the answer would be no. The answer for that would be, as soon as you add yeast to the tank, you only aerate then. So to give you an idea for how, I can say at least how a local macro brewery in the local area works. They have a rule where all their fermenter fills are only 3. So if the tank is, say, 180 hectoliters, which is a large amount, 1 hectoliter is one barrel contains, was it 1.13 hex? That's correct? Yeah.
1.2, yeah.
They will only cast in, so their brew house length is 60 hex. So it takes 3 worts to fill that tank, 3 cast outs to fill that tank. They will add yeast to the second one and aerate to the second one, because by the time they cast out their third one, the yeast cells haven't started yet. And this is the important thing you also have to keep in mind with this. If I'm able to, say, get 4 worts into that fermenter before the fermentation really takes hold, I would probably aerate on the first one. So it's not just, the time it takes to fill the tank is very important. Now, if, say, on day 1, I am filling that tank, and I'm not going to add another wort until day 3, so I have 48 hours or so inside of there, I'm only going to aerate on that first fill, let the yeast cells start ripping, let the yeast cells start going, and then not aerate on the last one. Because if I do, I'm asking the yeast cells to go from aerobic, anaerobic, and that's going to stress them out a good amount. Now, if I'm able to fill that tank in a single day, add everything to the first one, you can probably add more oxygen to the later cast outs, but it's not going to be a big problem. Not going to give you any benefit. What you really don't want to do, and we've had a few clients who have done this, is you never want to aerate your wort if there is no yeast present. And there's a few brewers that have done this. And the reason we aerate the wort is not to aerate the beer itself. It's to provide yeast present. There's no real benefit for us to be adding oxygen to that beer. If anything, that's actually going to cause damage to the beer, because all the hop compounds that are very sensitive to oxygen will now all be oxidized. A lot of delicate malt flavors inside of that beer will now be oxidized. You only want to be adding oxygen when you have yeast present. And if you look at some of the beer guys, they won't aerate their wort, they'll aerate their yeast. So they'll aerate their yeast brink. They won't aerate their beer. I think that answers the question. There's a lot, like it's not a simple question. The big things you want to make sure is you never want air to hit beer that's not yeasted, and you want to make sure all the wort can get in that fermenter as soon as possible. And if you have long delays, then you start getting into some really gray territory where you see strain specifications and things like that come into play.
Yep, I think that's a good answer. And yeah, like I agree with you that this could be a whole discussion in and of itself, talking about wort oxygenation and why that matters and why that can be the key to improving quality and consistency. Before we proceed, Nate, I'm wondering if you could hide the slides and show our faces. We had someone asking.
Oh, yeah.
I don't know why, but someone asking to see that. There you go. Okay.
There you go. I don't know why you guys want to see me, but I'll take it as a compliment.
Absolutely. Okay, so our next question, and I think this is also a good question. Any thoughts on glycerol production? It's hard to find information about control of glycerol production. Is it possible to control it during fermentation?
Great question. My understanding on glycerol is, or I think it's fair to say that the field's understanding of glycerol is minimal. We know that certain strains start producing glycerol to much higher degrees than others. Things like saisons and kveik's produce a much higher glycerol content than other ones. And from a saison standpoint, the higher temperature we ferment, the more glycerol we seem to see in the final beer, or at least greater perceived glycerol to see in the final beer. I actually don't know if it is a tangible increase. I don't know of any way that we can actually push it one way or the other than fermentation temperature, which would be higher. Now, there are other issues with fermenting at higher temps, which we'll talk about on Monday or the next episode of this webinar. Yeah, that's about as much as I can answer that on. It's not known fully, to my knowledge.
Yeah, I agree with you. We've been part of some research that's been, luckily, we've been able to measure this glycerol production in a few different projects. And yeah, one thing that is clear is that it is very much strain dependent. Some yeast strains produce a lot more glycerol than others. There used to be some trends associated with yeast families. And we'll get into that when we talk about yeast strain diversity in a couple weeks. And then the other part of that is that temperature does seem to play a role. And in general, hotter temperatures do tend to sort of push that metabolism towards glycerol. Yep. Probably because the cells might need it to actually protect themselves.
Yeah, there's also that, kind of high throughput thing, but we'll talk about that on Monday.
I think we already discussed this question, but the question was, is it useful to open ferment at the homebrew level? And I sort of chipped in there too a little bit.
Yeah.
Saying that, yeah, it can be pretty interesting and you can get pretty different results. I did an experiment about a year ago comparing open ferment with kveik versus closed ferment, and really preferred the aroma with the open ferment. And since then, for those yeasts, I've sort of done an open ferment for the first little bit, and then closed it up and found that that's helped with some of the aroma production. I think it's beer style, but
I've only done this experiment with personally with English style ales, using the equivalent of our English 2 strain, and I found that when I had a glass carboy and then plastic bucket open fermentation, and the open ferment one was massively more flavorful. The hops really shown in the carboy one, but it kind of had this really nice balance of kind of more fruity characteristic and hops in the open ferment. Now, one big thing with open fermentation, because I'd feel bad if I didn't mention this. You should not finish your fermentation in an open vessel. So the way in which all those large-scale breweries use the open ferment is that they transfer the beer into a sealed vessel near the end of fermentation. So if you're using, say, glass carboys, you would transfer to your glass carboy near the end of that open fermentation. You don't let it finish inside that ferment, because all that oxygen that's in the environment, once the fermentation is done, it's now just oxidizing the beer. It's damaging the beer. But during fermentation, it has some positive benefits to it. So there's an extra thing you have to add in when you decide to open ferment, but I would argue it's very much so worth it if you're set up to do so.
Absolutely. The next question, and that was a great question. If sugars are all consumed after 90 hours, why is it suggested to wait 2 to 3 weeks before moving on as a home brewer?
Because old homebrew knowledge is really hard to shake away. That's kind of the Coles note. So a few things I want to point out. Fermentation time is highly variable. So if I have a wort that is mainly composed of simpler sugars, that fermentation is going to occur much faster. Now, if I ferment that beer colder, the fermentation time is going to be elongated because it's going to be slowed down. If I use a kveik, I can ferment that beer in like 36 hours and it's done. The sugar being consumed is only one of the few indicators of doneness. That whole idea of wait 2 weeks is really it's consistent, but it's not a good standard, and it by no means works for pro brewers. Or if you are doing this professionally, you are wasting large amounts of capacity. And believe me, stainless steel tanks are expensive. You shouldn't be doing that. What we mentioned before about being the forced diacetyl test, the main reason for that 2-week wait period is that's how long was thought to be required to do for the diacetyl to be cleaned up. And that has a long history in homebrewers doing some weird stuff and supplies not being great. Like if you're only using dry yeast in the 80s or early 90s, I would be doing that because your yeast cells you work with, ingredients you work with aren't the highest quality. You need that time for it to kind of mature and age. But with modern ingredients, you really don't need that. And so I'm going to get back on track with the forced diacetyl test. From a professional standpoint, the 2-week standard is useless. Because you don't know after that 2 weeks if everything's actually done. We recommend the forced diacetyl test be used what's called a go-no-go test. So if this test clears, you're good to proceed to the next stage, which for commercial brewing, even for home brewing, is cold crashing your beer. So if you're, say, kegging your beer, moving that beer from ambient temperature into, say, 4 degrees Celsius. That way you can you get the process going further and further. Now, the issue here is that if I have diacetyl still left in my beer and I cold crash it, the yeast cells are no longer active. Therefore, they can't reabsorb the diacetyl. That's why the forced diacetyl test is used as a go-no-go test for that stage. Now, if you are doing the 2-week thing professionally, and I know this question was posed from a homebrew standpoint, but we see this being done professionally. And it just blows my mind that this is still a practice. I can get a fermentation done in say 1 week. And if I know that beer is completely clean and ready to go, I can now, just by getting rid of this 2-week rule, I can now put 3 times as much beer through my brew house or through that fermenter, which is a massive gain, like massive bottom line improvement. I don't have to buy 2 more fermenters. I just use the fermenters I got 3 times as fast. It's phenomenal. And also when you're storing beer in tanks, like I've been in breweries where, the beer is near the end of fermentation and we realize at that point that there's a leak at the top of the tank. So if I cold crash this tank, I'm going to pull in so much oxygen during it that it was usually IPAs. It's all going to be shot. So I've been on there on a ladder with like C-clamps trying to like clamp down a lid so that no oxygen gets in. If you just let your beer sit there for 2 weeks, because you're increasing the chance for oxygen to get in that beer and ruin it. Don't do that. That's what I hope I answered the question. I think I did. But that always bugs me. That topic always irks me. And if you want to learn more about this, by the way, you can refer here, but we also have a blog post at escarpmentlabs.com that goes over the forced diacetyl test. And it goes a bit more in depth. You can see more of Alex's lovely face in there. It's wonderful.
Yep. No, I totally agree with you on all counts. I think even if you're a home brewer, it's always worth, pulling a little bit of that wort and ultimately wasting a little beer to get more data on your fermentations and know when they're truly done, because you might save a lot of time. And especially for the beer styles that are really sensitive to oxygen, like IPAs, you're going to have better quality if you're able to move that along faster, as long as you're able to clean up any of the off flavors like the diacetyl.
Absolutely.
Yeah. So our next question is, I've heard underpitching can decrease Fusil alcohols and increase ester production. That seems to counter what you're suggesting. Is there any good that can come from underpitching?
I'm going to save that to Monday. That's a question that's perfect for Monday.
No, I totally agree.
No. My gut is no, but there's lots of controls we have to talk about on that. So that's not a total or fair answer.
Yep. And I may have a different perspective even. I know there are some yeasts where underpitching can be beneficial, right? So
yeah, we'll talk about a better way to conceptualize pitch rate, and we'll be on the same page when we get to that.
Sure. I think we'll just go for one more question and then we'll cap it off and try to save the rest for the Q&A session. This is a great question, I think. We really like the sulfur character in certain lager styles, Pilsner, for example. Are there any tips to stimulate sulfur production and maintain a certain level in the final beer?
Okay.
This is a question I'm personally passionate about.
Yes. I have a little bit of an interest in this as well. So there's whenever you're looking to control any sort of compound, and this is a really good segue into next week. This is a really good segue. And it kind of feeds in this week as well. You have two options. You can either maximize its production, or you can maximize its degradation. And so for sulfur is a good example for this. So when we're trying, I'm getting a little bit ahead of myself here. Just need to organize my thought. There we go. The fermentation is going to produce sulfur no matter what. Like that's going to happen. Like you almost always see sulfur being produced during a fermentation. However, sulfur often leaves the beer very quickly. Now, if you want to preserve the sulfur profile in a beer, open ferment probably isn't the way you want to go. So the enemy of sulfur is oxygen. It's absolutely oxygen. Like in a lot of stress, and you see this in a lot of stress ferments. Like look at some certain wine fermentations, mead fermentations especially. You see lots of sulfur being thrown off. And part of the reason they open ferment these often is so that the oxygen can hit that sulfur and break it down. And you kind of know this in your daily life. Often sulfur compounds are used to help stabilize food. They're help prevent staling when it comes to like juice boxes and things like that. Sulfur is everywhere. It's used to help mitigate oxygen stress. So what you can do if you want that sulfur profile, you can look at trying to maximize it. But I would probably first guide you towards trying to maintain and keep all that's in there. So as the ferment is going on, you can do a few different things. The most common one you can do is called spunding, which if anyone doesn't know what that is, it's a pressure relief valve you put on the vessel. So you can't do this if you're open fermenting, obviously. You should not do this if you use a glass carboy because that's how you make a bomb. You need some sort of stainless steel, or if you want to go really old school, wooden vessel that you can seal up. So near the end of fermentation, say 2 to 4 degrees Plato left, you can put this spunding valve on, and then the pressure will build up inside of the tank, and then once, say you set the spunding valve to 12 psi, once the internal pressure hits 12 psi, it will release, say the inside hits 12.5 psi, it'll release a little bit. I noticed a few people, just as an aside, in the chat talking about lagers, and mainly Skara Kveik or the kind of quick Kveik lagers. This is a great way to lock in that sulfur profile if you're trying to make a quick turnaround lager like this. Add a cap. You can either cap the beer, like you can transfer it into a corny keg, say 2 degrees Plato before FG, seal the whole thing up, and that way any sulfur that's being produced will be trapped inside. Now, if you want to go extreme on sulfur, some commercial brewers will essentially spund the entire way through ferment, which means that during the bulk of fermentation, they're always releasing this. Like they're always under pressure. This has a bunch of benefits because it means that there's a higher CO2 content inside the beer, which means that if there is a leak, if you do have that small seal where oxygen could get in, the pressure inside is enough to push out. So you have CO2 loss opposed to O2 egress. The other big thing you can do with this is ferment cold. Sulfur is a gas. It's gaseous. And if you look at anyone who's carbonated something via with a CO2 stone or with a fridge with corny kegs or things like that. The colder you bring the kegs, the more soluble those gases become. So if I ferment the same beer underspun at, say, 10 degrees C and 18 degrees C, that 10 degrees C is going to retain so much more sulfur because the sulfur is much more soluble at that colder temperatures. While the 18 degrees C one, a lot of that, the sulfur just isn't soluble at those temps, so it's all going to flash off. Even though when it's spunded, those flavors will flash off more so than they would at lower temps. Now, if I use that 18 degrees C as an example, I did a spund versus no spund. The spund would have more sulfur, but it's going to have significantly less sulfur than the colder fermented one. As a larger portion of this, part of the reason why lager beers often have a sulfur flavor to them is because they're fermented colder. If you add a normal ale fermentation temperatures, most of the sulfur is so minimally soluble that it's just going to flash off. But when we start using lagers to ferment colder, or even kolsches, the solubility increases, the temperature goes down, and we're able to lock in more of those sulfur compounds during the ferment. I think that's my full answer on that for now. But got anything to add?
No, I totally agree with that. I've been homebrewing lagers for a little while, and always found that it was hard to retain that nice, lagery sulfur character that really needs to be there for the flavor balance and the crispness until I started spunding towards the end of the ferment, and that made a really big difference. Being able to really retain some of those gases at the end of the ferment, and even use some of that pressure to help reduce any chance of oxygenation, especially with homebrew equipment. Like horny kegs are horrible for leaking and stuff like that. And then being able to use some of that pressure to also help along with transferring it off the yeast really helped with stability and quality of my homebrewed lagers.
Yeah. I would point one other thing out for this, and this is from a kind of a commercial standpoint. So you see some breweries where who use spunding, and their lagers are, I'd say, more traditional. They're not they're more of the Pilsner or Cal Warsteiner, kind of that old school German style or European style lager. If you look at the macro guys, they're not spunding, or they're not spunding nearly the same degree, and that's part of the reason why they're elbow bended. They don't have that sulfur profile. A lot of brewers will do a technique that we're going to talk about next week, which is called free rising, where essentially at the end of ferment, you increase the temperature of the beer so that it finishes faster. Yeast cells like higher temps. The downside of it is that you lose all the nice sulfur compounds. So if you want a really quick, clean, like elbow bending beer, free rise is the way to go, because it's going to remove it's going to scrub off any of these compounds you don't want. If you want them, don't do that.
Absolutely. Yeah. And I agree with that too. I found that I've been getting better lager flavors from slightly colder ferments. Like some people go super cold, but like even just keeping it around 12 Celsius and maybe get it up to 14 or 15 at the end to clean up the diacetyl seems, for me anyway, seems to work a little bit better than really, really ramping the temp aggressively.
Yep. I'd agree with that.
So yeah, low and slow. Cool. I think one of the things we've been talking about is having a full webinar on lagers specifically, and trying to find a guest with a lot of experience to help us out with that story too. So we're working on that one as well, and hopefully we'll have some news to share.
Like I said at the top of this is the introductory series. This is the thing that opens up all these different topics. Like we are planning all the different downstream, conversations about this stuff, because there's a lot to learn. Yeast is not a simple thing. This is the basics. This is not it's not everything by any means.
Oh yeah. We're still learning, right? There's
exactly.
It's a pretty complicated world. Cool. I'm going to cut us off there. I do see all of the questions, and there's a lot of good questions being asked as well. We're going to try to collect those and put together some answers in our Q&A session. And I'm really looking forward to seeing everyone on Monday coming up, and diving into part 3, where we're going to be talking about yeast flavor control and all of the practical things we can do as brewers to control the flavors in our beers and really get those results that we're looking for.
Yep. Look forward to it.
Thanks, Nate.
All right.
And thanks everyone for listening in.
Take care, everyone. Stay safe.